We are back, and we are still kind of in the middle of penicillins and the beta-lactamase family. We've seen what a penicillin is, and we've seen what it does, what all the penicillins have in common, and what's different between them. We saw an enzyme that can break a penicillin. We saw the penicillinase enzyme, and we saw an inhibitor that breaks the penicillinase enzyme. It is hard when you're learning antibiotics to keep this all straight, and that's why I make my face-to-face classes play this game, where we say yay if we're looking at something that is good for team humans, and boo if we are looking at something that is good for team bacteria. So, whoops, what the heck? Okay, so peptidoglycan, for example, is a team bacteria thing, so we would say boo. Penicillinase breaks our antibiotic penicillin, so it's boo. If we inhibit transpeptidation, that process of inhibiting transpeptidation, that's yay, that's what penicillin does. So this is a silly thing to do, but it's hard enough that typically classes, when they first encounter this, they get almost all of them wrong. So I highly recommend you go through all the words you don't recognize in the last 10 slides and try this. Okay, so a thing that I haven't talked about much is what happens in a patient when they take a penicillin, and it turns out that a lot of people have allergies to penicillins. So that's yet another thing a clinician has to think about. And somebody who's allergic to one penicillin, you have to assume they're allergic to other penicillins as well. And some of them are even going to be allergic to cephalosporins and carbapenems. So again, those are things we have to think about. And I want to come back to MRSA, methicillin-resistant Staphylococcus aureus, and its mutant transpeptidase, because again, this is the transpeptidase enzyme as drawn by a generous person who gave this image to the public domain. And this is the way we think about penicillin, or a beta-lactam. And the idea is penicillin reacts with the transpeptidase enzyme and poisons it. But MRSA has a mutated version that does not bind to beta-lactam antibiotics. So it has a few mutations in it, just the right mutations that change its structure until it can just ignore beta-lactam antibiotics. And remember, most of the antibiotics we use are beta-lactam antibiotics. So this is a major problem. And if you read the book Clinical Microbiology Made Ridiculously Simple, during the discussions of antibiotics, a lot of antibiotics are labeled as yes or no for treating MRSA. It's that big a problem. So that's one of the problems, but we still have a lot of other pathogens that are sensitive to penicillins. So streptococcus in some cases will be sensitive to penicillins. Other bacteria might be resistant to penicillins, but not to cephalosporins. And again, this is the biggest group of antibiotics. This is, I think, half of antibiotics that are prescribed are cephalosporins. If you look down here, cephalosporins have a beta-lactam ring, but everything else they have is different. So this beta-lactam ring is what does the work by binding to the transpeptidase enzyme. But cephalosporins have two regions that chemists can modify to make different versions that will interact with cells in different ways. They will all react with the transpeptidase enzyme, but they'll get into different cells. They will accumulate in different parts of the body, depending on what different changes we make here and here. So most of these are going to be resistant to penicillinases, and that's great. But there are cephalosporinase enzymes that break these. So that's no good. And MRSA is resistant to all cephalosporins. So here's a long list of cephalosporins. And the textbook skips the 5th generation. There are 5 generations of cephalosporins. What I'd say is, if you know these bold ones, that's all the cephalosporins you need to know. And on this slide, this is another one of the visual mnemonic devices from clinical microbiology made ridiculously simple, where they show the difference between the different generations of cephalosporins. And what they show is little microorganisms with little daggers falling on them. And a first-generation cephalosporin would not be much of a threat to a gram-negative such as E. coli, but it would be a big threat to Streptococcus pneumoniae and other gram-positives. Second generation might be somewhere in the middle for all of them, and the third generation would be weighted more towards the gram-negatives. So third-generation cephalosporins you might be able to use against E. coli. But not so much against something like Staphylococcus aureus. So it is complicated, and you do have to know every single one of them and what they work against if you're in the business of prescribing these antibiotics. So as bacteria become resistant to cephalosporins, we need yet more types of antibiotics, and carbapenems are some of the best. Imipenem, this one here with its beta-lactam ring, but it's very different other structures, has the broadest antibacterial activity of any antibiotic. So this is your broadest spectrum antibiotic. It's a kind of a backup, last line of defense kind of thing. And there are other carbapenems that are very similar to each other. So I don't think imipenem itself is the most commonly used, but still this is how to think about them. These are some of the best beta-lactam antibiotics. There's tricky pharmacokinetics and pharmacology with imipenem. That doesn't matter for you for this class. What does matter is that they don't work against MRSA. So that's a common theme. And then the very last one, the monobactams, there is only one, and here's its beta-lactam ring, and here's the rest of it, as trianam. And it only works against gram-negative bacteria, so it's the opposite of the original penicillins. And so if we mix this with something like vancomycin that targets gram-positives broadly, you get a broad-spectrum mixture. The enzymes we're going to see on the following slide are going to give bacteria the ability to break this beta-lactam ring and the beta-lactam ring in carbapenems. So these are the two enzymes we are concerned with. So bacteria that have these enzymes, they are resistant to carbapenems and monobactam. So we call them carbapenemase-resistant Enterobacteriaceae, because for whatever reason, the bacteria that have these enzymes are members of this bacterial family. And they include Klebsiella pneumoniae, E. coli strains, Enterobacter strains, Pseudomonas aeruginosa. And if these are carrying a carbapenemase, they are very difficult to treat. Because our first line drugs are cephalosporins. Well, these enzymes inactivate cephalosporins and penicillins and carbapenems and aztreonam. So these two enzymes are one of the scariest things, period. So this is KPC2 is the famous Klebsiella pneumoniae carbapenemase. NDM1 is the New Delhi metalloprotease, which does essentially the same thing. That doesn't matter to you, but you should know what CRE is. You should definitely know what carbapenem-resistant Enterobacteriaceae are. And so if someone wanders into your clinic infected with one of these bacteria, you can't treat them with any of your first-line drugs, and you don't know it until you try. until you either get lab results showing what the bacteria is resistant to, or you try to treat it with beta-lactams and fail. You don't know. And that's the worry. And that's what's happened in some cases in the past, is a person has come into a clinic with an infection. The first line drugs fail to treat it. The second line drugs fail to treat it. So then what? Well, there are a lot of different antibiotics. If the clinic had every known antibiotic in their refrigerator, they could come up with something that would work, but they don't. In a clinic, what you're trying to do is treat the most common illnesses as quickly as you can, and when you get something rare like this, you may or may not have access to the backup medications, the rare antibiotics you would need to treat this patient. So the question is, do you figure out what's going on with this patient and get the rare backup antibiotic in time to treat that patient? And in some famous cases, the clinic hasn't. By the time they figure out what antibiotic to use, the patient is dead. So that's what we're worried about with things like MRSA and things like Carbapenem resistant Enterobacteriaceae. And you'll see this in the news occasionally. Sorry, so I'm struggling with animations in PowerPoint today. What are you going to do? So that's the beta-lactam antibiotics. They all inhibit the cell wall synthesis. Vancomycin does the same thing. Vancomycin is this huge molecule, and it works against gram-positives. It's interesting because it's not absorbed by the gut. So if you want it to go into the tissues, you have to inject it. If you want it to go into the gut, you can give it orally to go to the gut. Vancomycin works against a lot of gram-positive bacteria, and it's very useful for that. It's used preventatively during major surgeries. And one of the problems we have is enterococci. So bacteria of the genus Enterococcus have developed resistance to vancomycin. And those weren't scary bacteria. They didn't cause a lot of infections until vancomycin became a popular antibiotic. And then those bacteria could compete against the others. And then Staphylococcus aureus, through horizontal gene transfer, developed vancomycin resistance. So MRSA strains could get the vancomycin resistance genes from vancomycin-resistant enterococci. And we'll see in the following slides what vancomycin resistance does. But I want you to understand that it hasn't become widespread, and that's partly because vancomycin-resistant Staphylococcus aureus can't compete against MRSA. Even though it has resistance to all these antibiotics, it's hard for it to maintain that resistance, and it grows slowly because of that. And so other bacteria can outcompete it unless vancomycin is present to kill off all the competition. So if a person has vancomycin-resistant Staphylococcus aureus, and they treat it with vancomycin, those bacteria will win. But if they treat it with anything else, they won't win. So a much more common thing is vancomycin intermediate Staphylococcus aureus. And these can be treated with a higher dose of vancomycin. They have an extra thick peptidoglycan layer. And compare this extra thick peptidoglycan layer to what I'll show you in a couple slides. So I'll come back to this. A key point that a lot of students miss is that antibiotic resistance is not the same as virulence. It is not the same as pathogenicity. So if we are scared of MRSA because it is resistant to a lot of antibiotics, and we're scared of Carbapenem-resistant Enterobacteriaceae, and we're scared of vancomycin-resistant Staphylococcus aureus, we're scared because we can't treat it. But that doesn't mean those bacteria are going to go out and attack healthy people, necessarily. So in the case of VRSA, those are not highly virulent bacteria. They lose their ability to cause disease by gaining vancomycin resistance. So you have to keep separate in your mind antibiotic resistance versus the basic ability to cause disease. So if a bacterium can't cause disease, it can be resistant to any antibiotic under the sun and we wouldn't necessarily care about it. It's when bad pathogens get antibiotic resistance, that's when we lose the ability to treat them. So our worst nightmare would be something like plague having antibiotic resistance, anthrax having antibiotic resistance, because those could be used in a terrorist attack and we wouldn't be able to do anything about it. So that's what we would be afraid of in terms of antibiotic resistance. Any clinician wants to be able to treat your patients, right? You want to be able to treat whoever comes into your clinic. And so you don't want any antibiotic resistance to exist. But just remember, it's not the same thing as virulence or the ability to cause disease. So how does vancomycin work? Well, remember what a healthy cell is trying to do, what Staphylococcus aureus, for example, is trying to do, is the transpeptidation reaction. So this transpeptidase is trying to basically remove this amino acid and form a bond between this one and this one to cross-link these two amino acid chains. But yeah, if this step fails, the cell doesn't know about it, and it will lyse. And that's what any beta-lactam antibiotic is trying to do. It's trying to poison this. It's trying to prevent this reaction, and the cell won't know it, and the cell will lyse. So how does vancomycin work? Well, this would be the normal situation for a healthy cell with no vancomycin. If vancomycin comes in, what it does is wrap around the amino acids so that the transpeptidase enzyme can't bind, and it won't be able to do the transpeptidation reaction. So think of vancomycin as Pac-Man that's going to bite the peptide chain and prevent transpeptidation. Vancomycin resistance changes what was an alanine amino acid to a lactic acid, and vancomycin can't attach to it very well. So vancomycin falls off and the cell is able to go ahead and do that reaction. So vancomycin resistance physically alters the peptidoglycan in order to prevent vancomycin from working. And that's why it has a fitness cost. Vancomycin resistance slows down the bacteria and makes them more likely that our immune systems can defeat them, because they have to go through and change all their peptidoglycan. Vancomycin-resistant bacteria don't maintain this all the time. They sense the presence of vancomycin, and as fast as they can, they remodel all their peptidoglycan to look like this, to have this lactic acid instead of the normal alanine amino acid. And that is kind of poisonous to them, but it's better than vancomycin. So again, practice that yay boo. This stuff is hard. I'm sure that I lost a lot of people in this particular video, so go back, take it slowly, and do your yay boo so that you understand everything on these slides. And when we come back for the next video, we're going to look at all the other antibiotics. And you'll see there's not a lot of detail you have to learn. I'm not going to make you answer a lot of questions about specific antibiotics. So we'll see these in the next video.